Good evening and a warm welcome to the all participants in Sri Lanka. It is pleasure to have you join us Astro Week signature event under the Eco Astronomy Inc Astrobiology and Paleontology program. Today's sessions are detecting undetectable DNA sequence and its implication for the search for life. So we are going to journey into the explore the cutting edge intersections of the molecular biology and the planetary science. We are honored to host our guest speaker, Dr. Jyothi Basapathi, a senior research assistant in astrobiology at Northumbria University, United Kingdom. Dr. Basapathi brings an extensive academic background in biotechnology and molecular science, have earned her PhD from University of Adelaide. Her research specializing in assessing the limits of life detecting in extreme environment, work vital privilege to the planetary protection, spacecraft monitoring, and the ongoing quest to identify extraterrestrial life. Currently, she is a key researcher within the planetary science and astrobiology group at the Northeast Space Technology Center. Her expertise in biology, signatures, and the DNA detecting provides a unique lens through the which can understand how humanity might identify life in the most challenging corners of the universe. We are privileged to have her share those profound insight with us today. With that being said, I would like to invite Dr. Jyothi Basapathi to address Eco Astronomy International Platform. Dear Dr. Jyoti, now it's your time. You can get started.
Hi, thanks for the introduction. Hi, everyone. Just give me a minute. I'll share the screen.
Yeah,
Yes, thanks for the introduction and this opportunity. It is nice to
yeah,
always engage with students who are looking forward to exploring these fields and introducing to them about astrobiology or any field as such in science. It's always nice opportunity. So today I'll be introducing you all, like he said, a little bit about DNA sequencing and how detecting it would help for search for life both actually on Earth and extraterrestrial. So, like he said, I'm Jyothi and I'm currently in Northumbria University. And a little bit background about me, I did my undergrad in biotechnology back in Bangalore and India. And then I moved to Switzerland for my master's and I pursued molecular life sciences. And the duration between my undergrad and master's is when I wanted to see, is there something between space and biology? I didn't know what is astrobiology that time. So I just was looking up and Googling, and that's when I just found astrobiology. And I was like, okay, wow, this is interesting. Let me see more about it. And I got opportunities to attend some conferences when I was in Europe that time doing my master's. So I went and attended one of the conferences. I learned more about astrobiology. And then I landed for my PhD in University of Aberdeen, which is in Scotland. And I did my PhD. And now with the same team, we are all working in Northumbria which is in Newcastle in England. So our team moved from Aberdeen to Newcastle for this particular space research institute, which is newly established within our campus. It's called Northeast Space Skills and Technology Center. It's a new one and it's going to be starting this year. There's lots of collaborations between academic and industry to equip the students with a lot of space skills and technologies to provide them undergrad and master courses that is exclusively within the space sector. So we all moved here and we're working as NEST team members at the moment. So which is why we're in Northumbria right now. And before we all begin, like just a brief introduction of what is astrobiology. It's actually not as small as we think. It is a very nice place where it brings a lot of scientists together. Like it's very interdisciplinary. The one thing I like about it is how it brings biologists, chemists, geologists, physicists, engineers, everybody to answer the same big question about life outside our planet, but focusing on different areas of life. For example, some of them focus on understanding origins of life, some of them could be evolution of life, and then whether it is distribution of life on Earth or outside Earth, and what is the future of it. like we can see how there are different missions already ongoing, and these are specifically for NASA, for example. OSIRIS is in busy understanding about asteroids, or James Webb Telescope is looking into images of habitable planets and looking for planets that are having habitable environments. And then we have Perseverance and Curiosity rovers that are exploring Mars environment and understanding how Mars is like, or what are the samples like. And currently it is on the way, Europa is on the way to Jupiter's moon to understand the oceans of Jupiter's moon. So like this, there are various missions that are ongoing. And our team, particularly me, we are more interested in life detection. And for this life detection, what do we look exactly is something called biosignatures. So what are these biosignatures? They're just basically any biological components or that hints us directly to life. It could be DNA, it could be proteins, it could be a cell as a whole cell, or it could be atoms, it could be even fossils, or even patterns. Like some of you may know that there are like riverbed patterns that are identified on Mars. So all of these could be indicating as a potential biosignatures. And we use different models of biosignatures. They could be like microbes, or they could be some gases as well, organic gases, microbes, and archaea, viruses, biotic components, and even tardigrades. Tardigrades are like, they look like little piglets, but they're tiny organisms and very resistant. So we use all of these to understand biosignatures. So what's more important for a biosignature detection is that it should be sensitive, it should be reliable, it should be an actual signature, because it could be very confusing, these things. For example, if it's coming from contamination instead of an actual biosignature, it could be considered as false positive. Or if we have some sort of biosignature existing in an extraterrestrial sample, but we don't have enough instruments or techniques that are sensitive to detect it. So it should be reliable, compatible, detectable, and scientists have come together to classify biosignatures as something which is like a worthy biosignature, which is definitively a life indicating signature, or it is like an abiosignature, that is possibly something coming from a abiotic process, not that is something natural, or an ambiguous biosignature, which means like they could be in between biotic and abiotic. For example, methane gas. Methane gas could be something from an organism, like methanogens produce methane gas, or it could be something just a process that produced a methane gas. And agnostic is another interesting thing, which is quite debatable, where it's not something we even know defined within our knowledge at the moment on Earth. So we know how life looks like on Earth, but there could be something completely different which we don't know. So there are different forms of biosignatures. And as a biologist, I'm more interested in DNA, which I'll talk a bit later. But what are like the different techniques we use? There's so many techniques detecting these biosignatures, like microscopes, spectrometers, or spectroscopies, or fluorescence techniques, or like sequencing techniques, which I will also be focusing on, the nanopore sequencing. Or there are future techniques, which is like mostly now AI integrated biosensors, which is picking up more sensitivity towards these biosignatures. So as a biologist, for my research, I'm more interested in microbes and DNA. And we all know microbial cell, or any cell, is mostly made up of water, which is why when we look into any planet, we first focus on looking for signs of water. And then there's rest is like different forms of chemicals, which is like DNA, RNA, phospholipids, proteins. All of these come under the category of biosignatures. And I'm more interested in DNA, which is a double helix structure, which is a part of all our cells, microbial cells, and hereditary material. It is quite stable because of its strong bonding, because it's double helix compared to RNA. It's quite stable also because of its backbone, sugar phosphate backbone. So I was interested to learn how DNA could be detected in very small amounts, if possible, as a biosignature. So to understand that first, I'll just give a background about what is extreme environments or why astrobiologists look into extreme environments. We know that there are lots of microbes are pretty much everywhere. They everywhere you go, there's microbes somehow detected, and they have mechanisms. They have evolved mechanisms that can thrive in really harsh environments. Extreme environments is something like a lake which is very salty, which normally life wouldn't exist, but there are microbials that have adapted to exist on these lakes. Or you can see these deserts and arid environments, which means there's barely any water and not enough nutrients, but still somehow they've adapted to survive there. Or very high temperatures or extremely low temperatures. So the microbes surviving in these are called extremophiles. Like I mentioned, there are different ones. Thermophiles are the ones that can withstand high temperatures. Halophiles are something that can withstand high salt concentrations. Xerophiles are something that can withstand low water availability and somehow still survive. And then there's radiation resistance. There are many more. But how does this answer our interest of astrobiology? So when we speak about understanding habitable environments, we want to define certain characteristics which we can say, yes, this environment can still be good for life to exist. And that knowledge can be obtained from understanding extreme environments and what are the parameters within which these organisms can survive. If you think, for example, we were doing one of the research, and if we understand as 50% of water availability is enough for a microbe to survive, when we did some research, we understood some of the microbes can go even more lower, like even just with 35% of water, they can actually still thrive. So this is how you do explore extreme environments and expand the knowledge of habitability. And when you extrapolate this knowledge into any extraterrestrial planet, when you understand the parameters, you can then predict if it can host life or cannot host life, and it's worthwhile exploring the planet or not. So but there are some challenges with the extreme environments because of how there is high temperatures or radiations and things like that. The DNA that is released into these environments usually get damaged pretty quickly compared to a very normal environment. Or when you have nutrient scarcity or low water availability, the cells don't thrive as much. Like, for example, when you do in a lab, when you put all the microbiologists would know, when you do these nutrient rich media and then you put microbes in there, and then within 2 days you see that they would have fully grown. But then when you go to these environments, because of scarcity of nutrients and water, they're very low. So extracting all these cells, collecting them together and extracting DNA, that is detectable DNA, would be very, very difficult and challenging in these environments. I've made a small graphical representation in the next slide as well. You can see these are like different low biomass environments. These are all extreme environments, and low biomass means like there's very little amount of cells compared to an actual proper environment. Say, for example, a desert is just per gram, you can find only 10 power 6 cells, or in aerosols, you can find like 10 power 5, or in deep hydrothermal vents, you can just find like 10 power 4. And in clean rooms, it's even more supposed to be very minimal. So if you see a graph, normally when you pick like an agricultural soil, you expect like good amount of cells and DNA. So per gram, you can say you'll get like micrograms of DNA or nanograms of DNA. And then as you go down, you can see how it's coming down and down and down, and it gets more harder. So normally, the techniques currently we have is very popular in detecting micrograms and nanograms of DNA, but I wanted to focus on picograms of DNA because all these low biomass environments is having very small amount of DNA. So we wanted to see if any technologies that are existing now can actually identify these picograms of DNA, which is 10 power minus 12 grams of DNA. And for that, I looked into nanopore sequencing. So if you don't know what is DNA sequencing, it's just reading the bases in particular order and then understanding it and getting the whole reference genome so that you can map it back to an existing organism and identify whose DNA it is. So we do that very famously in forensics or an understanding in diagnostics. Most importantly, you would have heard probably during COVID time how we were all doing RT-PCRs and all those things, and that's how you identify the COVID gene in your sample. So similarly, we focused on one of the sequencing technologies, which is nanopore. So they have like a protein membrane, which you can see in this image. And when the DNA structure, double standard DNA structure passes through it, the changes in the ionic current, which you can see in the computer there, will read your DNA. So this is how they identify the DNA strand. And the good thing about it is one of their equipments is very tiny. It's portable, so you can literally take it anywhere and do it on field as well if you want to. And another exciting factor, it's been tested in International Space Station as well. They swapped few samples and they tried it, and it did work. Of course, it's a very closed and nice environment in International Space Station, but the successful launch and working of it is still an achievement. So from my PhD, because I'm interested in picograms of detection, which I mentioned, which is like low amounts of DNA, one other factor I wanted to do was not amplify the DNA. So a lot of the times in research, what they do is when there is very little DNA, they amplify it first, which is like doubling the DNA amount artificially using some enzymes and everything. And when they get enough DNA, then they sequence it. So why I wanted to not use these amplification techniques is how I mentioned earlier about false positives. So sometimes amplification can lead to false positives because the components you use for amplification, like the enzymes and stuff, they can amplify themselves. And then you might think that is a signal from a sample, and which would not be the case. So to avoid all those confusions, we wanted to see no amplification and directly use the DNA as it is, whether it is broken, whether it is in the same length or whatever. So to use it as it is, that was my interest. And for that, our team built a clean room. So this is like a closed setup where I can work with minimal contamination, whether from outside or whether it is from within the samples. And there's very low particle amounts as well. And I'll wear the full PPE kit, so there's no source of DNA or contamination from my end, or we try to minimize as much as possible. So all my experiments was conducted within this clean room environment. So how did it do? So this sequencing, the company usually recommends around like 400 nanograms if you're not amplifying your DNA, or just 1 nanogram is enough. But what I did was modified some of the protocols, and then we just used some standard DNAs that's commercially available, basically from a bacteria or from a fungus, which is like a yeast. And we went to diluting the samples to low amounts. And then we went to the lowest, which is just 2 picograms of DNA, and it was successfully detected by the MinION sequencer. And we published the article as well on this. And part of this was also applying this to an actual setting. So you all know about the Martian simulants, even your research institute is making one. So we bought this MMS2, which is a Mojave Martian simulant from Mojave Desert. And we thought maybe they have some native microbes. They're not fully, it's not a fully sterile soil. So just put some water in there, incubate it for at certain temperature for if any native organisms are there to, grow if they want to. And then few days later, I extracted some DNA and got 600 picograms. 600 picograms is still not considered very high. It's still a low biomass sample. Usually I said it's nanograms or micrograms, but because the MinION was able to detect 2 picograms, 600 picograms is still good enough. So once you sequence and then you extract the sample, sequence it and analyze through some bioinformatics tools, you'll get a list of microbes this way. So that's how you know, like, oh, these are the microbes that are actually existing or not. This is how you understand. And you always also see a reference below Homo sapiens, which is mostly contaminant DNA. So this is what I meant by false positive. So you'll always have somehow human DNA that could either be because of me, or it could be because of the reagents that I'm using for all these processes, because these processes are not just straightforward where you extract DNA and you just put it. There's a lot of steps that goes in between, where you add different reagents, add different enzymes, you incubate them at certain temperatures and all those. So those reagents cannot guarantee DNA free or contamination free yet. People are still working on developing those, especially for space interest. But at the moment, those reagents still have some sort of contaminants, and that is called like a keto microbiome. So you can see some of them, like even I'll just mention Homo sapiens, and the other thing just above it, there's something called Cutibacterium acnes. It's actually like a acne DNA. You don't expect that in a Mojave. So that's definitely a contaminant. So this is how we try to avoid, or how we should, this is how we can learn how to avoid a false positive in an actual sample DNA.
Currently, NASA is planning a mass sample return mission where a lander is going to go and collect the samples where a rover is already collecting some samples. And this lander is going to go collect those samples and bring it back to Earth by 2033. When we get those samples back, we want to see if we can detect anything, DNA for example, in a very, very low grade amount. So the current existing technologies cannot detect very low amounts, but we explored this MinION sequencer if it can detect really low amounts, and we were successfully able to detect DNA at an extremely low amount, so which is a very significant, because now this technology can be further developed and be ready when the mass samples are returned back to Earth, so that we'll be able to detect anything. So we all know that living organisms is made up of genetic material, right? So we call it DNA. So if we find DNA on Mars, and then we can say that, oh, for sure, there was life or there is life. It is for the first time ever that we are going to have Mars samples. So we are hoping that there could be some detailed analysis and new discoveries that has never known before.
Sounds exciting, but unfortunately, the mass sample return mission is now cancelled. So this was during my PhD, and now USA has taken a decision to cancel. But the technology and the discoveries are not going to go waste, because there could be any other country coming up to, one day just go bring the mass samples or any other samples, and then definitely we are going to work on developing the technologies to detect DNA if there's any extraterrestrial samples that is returned on Earth. But now, just for your knowledge, the mass sample return has been cancelled. So coming back to my research, so the same technology we wanted to apply on a different low biomass environment, for example, like air. And our team again built a bioresol sampler, and you can see on top there's a filter put on it. So what does this sampler do? We just left it outside for 24 hours running, and then it draws air continuously. So when it draws the air, the microbes are sort of settling on the filter. So I take that filter and then extract DNA, and then I sequence it again. Same with the nanopore sequencing technology. And this is how, again, you'll get a list of organisms, which is like from the air. So this is how you can apply this technology to not just on a solid state, but a gaseous state, and also in liquid as well. So we, again, as a team, few years ago, we went to this Bulbi mine. It's an underground mine, which is in UK, and it has a laboratory down underground. And it had a briny pool over there. So we wanted to see if there's any microbes. Actually, it's an artificial pool. Interestingly, it's not an actual pool, but we just wanted to see if there's any microbes thriving in a very salty pool like that. And I did ask my colleagues to get the samples, and then same applied whatever technology we had, and then found a very interesting organisms, which, like I mentioned before, about extremophiles. So the ones that love salt are called halophiles. So you can see there are like halobacterials or halobacteria, haloarchaea, and these kind of microbes that are thriving in there somehow. I don't know how did they come to these underground laboratories, but they're just somehow there. So apart from all these, now you understood that how these technology or how this detection technique can be used in all three states of matter. But all of these had a definite length of DNA. So what if the DNA is very broken apart? Most of the techniques again cannot detect broken DNA, which is called like fragmented DNA. Like you can see here, radiation in space. We know space has harsh radiations. So radiations will cause DNA damage. Now, if the DNA is not of a certain length and if it's broken, how nanopore can detect or will it detect is what we wanted to check and see. So again, one of my teammates had, my supervisors actually team had these samples, rock samples that was collected from few analog sites. And they were powdered and put into these tiny bottles. And you can see that they were put in a radiation chamber and they were subjected to high doses of radiation. So then it was given to me to see the differences between samples that were subjected to radiation and no radiation. And we were surprised by the results, how we got detectable DNA even after radiation of subjecting them to high doses of radiation from these rock samples. And I won't go into details of the graphs, but you can see that when you compare both like 10 mgy, these are all the radiation doses. So if you can see both the ones that were undergoing radiation as well as no radiation, the DNA and the fragmented DNA could be detected. So that means it's a very nice advancement to see that this MinION is sensitive to both fragmented DNA as well as good length of DNA, which is important because when we expect samples in space, as we have the current knowledge, we think it was mostly broken DNA or, yeah, mostly broken DNA. So DNA sequencing, apart from like knowing about extreme environments, it has a lot of other applications too, just for your information. Like in health sectors, they use it, clinical studies and diagnostics, like the picture says here, in Mars samples returns or future space missions. And on your right, on the right corner, you can see some guy trying the MinION in Antarctica area, so very icy environments. And even to understand climatic change, like how the microbes are existing, what kind of microbes are existing in certain conditions, and understanding the diversity within our environment, what kind of microbes are thriving in extreme environments. And another interesting thing is planetary contamination as well. So for planetary protection, there are either forward contamination or backwards contamination. There are different methods to avoid contamination when there's a mission going on. And DNA sequencing could be another technique to identify if the instruments or the samples are clean enough to be launched, or backwards, either ways. So there's many applications of this technique, and we're excited to further explore and see how this is all going ahead. And apart from sequencing, just a brief introduction about our team, what they do. We have this Martian chamber as well built. Like I said, our team is fully engineers and physicists. I'm the outlier as a biologist at the moment. But it's interesting because you get to work with such exciting projects together. And this chamber can simulate Martian environments in terms of the humidity scale, in terms of temperature, pressure, radiation, everything. So I hope I can in future do some experiments. Currently, I haven't done anything, but I'm looking forward to do some of the experiments within this chamber. They also have an instrument called Habit, which they have been building for a long time now. They've been working together, and it's an instrument that was supposed to go to Mars. And it looks something like this. So it has two components, which is the environmental package and the bottle. And Habit was meant to, or is meant to, investigate liquid water on Mars, actually. And this bottle is the first Europeans in situ resource utilization instrument on Mars. And it was just to test if you can produce liquid water for future Mars. I have another video that will explain it a bit better.
This instrument was developed to operate on Mars. Since Mars is far away from Sun compared to Earth, the range of temperatures can be minus 120 degrees at night and up to 20 degrees plus in the morning. Because of the lower or thinner atmosphere, the temperature is not that equally distributed. There'll be up to 25 degrees of temperature difference between just the feet and the head. On Earth here, it's predominantly nitrogen and we have oxygen to breathe. But there, it's 95% carbon dioxide and very little moisture of water vapor and very little oxygen. So this instrument I have in my hand is called HABIT. It stands for Habitability, Brines, Irradiation and Temperature. It has sensors to measure temperature of the ground, temperature of the air, and also the ultraviolet radiations. The main goal of the instrument is to test the water cycle on Mars. So there's a hypothesis that during night the water vapor is absorbed by the surface of Mars and in the morning it evaporates back. But we haven't seen a physical proof for that. So we built an instrument where we can simulate the Martian surface. Right now inside the chamber it's about 6.8 millibars, which is within the range of Martian pressure. If we can produce water on Mars or air on Mars, even food, like you watched in the movie Martian where Matt Damon produces potatoes on Mars. So if we can do that as much as possible on Mars, then we can reduce the amount of things we carry from Earth. With HABIT, we are going to see how water can be produced. And if it proves successful, then the next step will be to produce it in a large quantity and to see how it can be useful for the astronauts who are going to go there in the future.
Definitely, I would like to acknowledge the whole team and Northumbria University, the Quadrat who funded my PhD, and Oxford Nanopore Technologies as well for their constant collaboration with us. So thank you so much. And of course, thank you to Eco Astronomy and Arundu for this opportunity. Please feel free to ask me any questions, not just about science, if you want also about career guidance or just understanding how to pursue astrobiology or anything, just feel free to ask me any questions. I'm happy to answer them.
Thank you very much, Dr. Jyothi Basapathi, for having very nice discussions and a very informative discussion. Dear students, now we are open for the Q&A sessions. If you have any further questions regarding today's sessions, and make sure to drop into the text to our chat box, so you can just raise your hand, then I can just call you one by one. So until I have a few questions to our young explorer. As an example, if we could have any opportunity to find out microfossils coming from the space, do we have any chance to detect that Asian DNA by using any kind of samples?
Yeah, yes, hopefully the sequencing technologies can detect ancient DNA, but if you remember, I mentioned about challenges with these, protection of the DNA. So there was, I think, one research, I think with Perseverance itself, they were more interested in understanding subsamples rather than surface samples. So we believe that its DNA may be more preserved underground because the radiations is a bit lesser. So if there are chances with that, I think, yes, you can still detect. There has been a paper, I think, where they analyzed almost like 2 million year old DNA, ancient DNA from fossils. But of course, there's more scope to further developing and understanding more.
That sounds much interesting, and also while we're just exploring the life beyond the planet Earth, with your experience, what kind of life that you are seeing, like same as in our planet Earth or something different? So what we are exactly looking for?
Personally, I think for me it's more interesting to see how the current microbes on Earth can exist. So the other aspect of astrobiology, which probably I forgot to mention, is in situ resource utilization, like how in the video you saw Dr. Miracle explaining about HABIT. So we want microbes also for different things on space. It could be for converting gases to production of like oxygen, or for food production or plant growth, or anything we need microbes. So if you want to see how these microbes can survive in actual Mars as well, you exploit the Earth current existing extreme microbes into, like, you put these in simulation chambers and see if they can survive and things like that. So that is another aspect of astrobiology, in situ resource utilization, which I'm also very interested. Or there's another, like you asked a question about life outside. So honestly, I'm not sure yet. So personally for me, I'm not expecting like a actual round living cell, microbial cell. But if there was any past life, because we might all know that at a point Earth and Mars had similar atmospheres. So if life evolved on Earth, then why did not evolve on Mars is one of the big questions asked. So if you believe that there is past life, you can even search for existing DNA that are preserved there. So I'm looking forward to look any DNA structures on samples.
Okay, thank you, Jyothi, for the answering that questions as well. And now moving to the our chat box, and I noticed one of our students have been just asking the one of question, and what is the process of encoding the data into the DNA? The questions coming from the Abiru.
That
Yeah.
I'm not very sure of how they plan to store data into DNA, but yes, but I haven't heard that into too much into space applications, but yes, there is some research I have heard about this of encoding the data into DNA, but I'm not very aware of the process of how DNA can be used for that. I know it is capable of a large memory, but how it is encoded within the bases, I'm not very sure of that.
While we are just go through with that same question, the life beyond the planet Earth. So do
Yeah.
we have any potential biosignatures from the outside then with the real samples? I think like you mentioned, like we have to just wait until mass sample retention. I know that many of our scientists are waiting for that good news, but anyway, so when will we receive some exact samples to confirm in like the life that we are just expecting?
Yeah, but if you see some of the I mentioned about Osiris mission, which was asteroid Bennu. So from that samples, they have already detected few nucleobases. So there's a difference between the base and then nucleotides and then DNA. Nucleotides is like a polymer structure, and for DNA to be formed, you need multiple nucleotides coming together. But Bennu asteroid was having some, actually all of the nucleobases. So adenine, guanine, we have 5 nucleobases, and uracil, all of them were able to detect. So that's one positive sign already. From extraterrestrial material, we were able to identify a few nucleobases. But with mass samples, still not sure. We are expecting also China. I'm not sure if that's really true. China is having a mission to get some mass samples, or we're hoping US itself would potentially have a relaunch of this back and say that no, we will have mass sample return mission back. Or if anything, I think if even the mass samples are not returned, if we could send out instruments that can, if it's going to Mars directly, a robotic instrument that can take the samples, analyze the samples, and detect anything. That is also still one way to go ahead. That it's long time, but robotic missions are already there. So just adding few detecting instruments. I think the current rovers, they have Raman spectroscopy or different spectrometers already detecting signs of organic molecules or gases. So why not something to do with DNA then?
And that sounds very interesting. And also, I'm moving to the another questions. What do you think, like preservations of the DNA while the meteorite entry? Do we have any evidences after the touchdown of any kind of meteorite, so without having the contaminations of other DNA?
Yeah, I think, yeah, like I mentioned about asteroids, I think there was another meteorite. I'm not sure about the name, but also they try to study organic molecules, and they're mostly interested in the organic chemistry part of these meteorites. With DNA, I think, except for the Bennu asteroid and Ryugu, there's no signs of nucleobases found so far otherwise.
So, like,
Yeah.
I noticed that while you are just presenting, there is a very nice chamber, the Martian chamber. So, is that innovations coming from your university or something like another institute?
It's our team itself, yeah. They built the chamber. They designed the chamber, and then of course they gave it to a company to build it, and they built the chamber. And yeah, we have the coolant, and we have all the vacuum suction as well, so that it creates low pressures. And yeah, so it's the chamber is fully ours.
That sounds really good. I also just looking for something similar. Like today, I noticed that you guys are working something because of that is kind of a much more a scientific analog. I believe, like in the future,
Yeah.
they can get something like much more proper data, and also like while you are just working with such a simulants. So, what kind of simulants that usually you are using? I noticed that I feel like it's a Exolith that you are just taking.
Yeah, so far I worked only with Martians, Mojave Martian simulants only, like the MMS1 and MMS2. I've never worked with others like JSC or JSC or MSG so far. I haven't. I think so far I feel the closest is Mojave, the one where they have added all the salts and yeah, being sold, I think, in Texas by Martian Garden. Not sure if you've heard about that one. Yeah, so
Yeah.
that's the one I've closely used for my research so far. I haven't used any other regulates.
Do you have any kind of dreams in your childhood, like in the future, that you are going to select in the astrobiology or biology? Or so, what is the motivation for just studying over there? Because of, I think, like you also know, around in South Asia region, we don't have that much educations and the outreach regarding the astrobiology. I know that very minimum groups are trying to develop this knowledge, including you. And what is your experience and why you selected kind of field?
Yeah, I think I was mentioning during my introduction where I said that it was accidental, but I was just interested in space, but I wasn't very confident in pursuing physics or that time the only knowledge we knew about pursuing space was probably either through physics or mathematics. You think either it's just astrophysics or astrochemistry. Nobody would have thought biology and space coming together. At least it wasn't very popular. So when I saw that opportunity, because I was interested in space, I thought, and biology together, I thought, okay, why not just try this mix and see what exactly they work on. And once I got in, I realized that you can be from any field, like even if you're just in a molecular biologist or just a chemist, you can definitely apply all your skills in space. You just need to have some knowledge and background, understanding. Like you can do just some online courses and understand the ongoing research, and you can always apply your skills to, it's all transferable skills. There's nothing called exclusive astrobiology skills. It's just transfer. I can even go to health sector tomorrow and do the same skills I've been doing, same sequencing and genetic study and all those, and I can apply the same knowledge in space sector as well. So that's how I thought, let me explore my skills into space field because I'm interested in space.
Also, I'm going to dig into the chat box and Zelina Sasmika. There is another question coming from one of our students. And Madam, can we understand the internal biological system of the living organism by examining its DNA?
Yes, definitely we can understand. When I mentioned about extreme environments and extremophiles, so when we study their DNA, you can understand how its mechanisms is modified. Why is it adapting, better than other organisms? Not every organism can survive harsh environments. So what is the particular sequence, or why everything is basically encoding to protein? So DNA gets transcribed to RNA, and then it's translated to proteins, right? So there's like, if you have learned in school, there's like transcription and translation processes of biological systems. So when we further analyze the RNA and understand what is the protein it's encoding, to protect itself from high temperature or low temperature, you'll understand the internal mechanisms and functions behind how it's going. So yes, everything starts with the DNA, understanding any internal biological system is through DNA.
That's something another questions. What is your experience regarding the theories like panspermia? So what do you think? Like, what kind of view that you are just looking for about panspermia?
Yeah, it is one of the theories that life could have come from outside. But if you ask my personal opinion, I'm not a big fan of that theory. I don't think it's coming from outside. It's just how Earth evolved and how it is, how it's been created in the end. It's just if our planet is special, it can be special. But there are many theories to understand evolution and to understand how a microbial cell started on Earth. But panspermia is not my favorite personally.
So I think like way of most of scientists moving with the panspermia, it's getting something like pseudo part. So that is
Yeah.
the reason, and because so we are just completely out of that current science, and that's why
Yeah.
I just want to personally ask you like what kind of
No,
view.
definitely. Yeah, because in schools, I think we used to all study back then a lot more about panspermia and everything. But as in more research is going on, you see how it is. All these theories are deteriorating.
Yeah, that's exactly true. And also, dear students, so we are opening for the Q&A session. So today we have already discussed about the DNA and implications and how the DNA we can just use for understanding life beyond the planet Earth as well as life on our planet Earth. And also, Jyothi, with your experience, like if someone interested to select a subject like astrobiology in the future, so what will be your advice? Like what kind of subject that many kind of students have to be enrolling in their school level before they head into the university level?
Yeah, that's very nice. Yeah. I think in school, you can just pursue basic STEM, just study everything, physics, chemistry, mathematics, and biology if you're interested in biology. But after school, when you go to undergrad, for example, if you see my background is again biotechnology. Like I said, all the skills can be in the end applicable to astrobiology. But now I think there's more scope. For example, in US and Europe, there are some planetary sciences courses or astrobiology courses. But my advice is usually to have an undergrad degree that is more generic, not so focused to just astrobiology. So have a generic undergrad degree. For example, if you're interested in chemistry, do something related to chemistry in your undergrad, or if you're interested in biology, then do biotechnology. Or if you're an AI person or computer, then of course you'll do engineering, computer science engineering, or communications. All my teammates, they have backgrounds in like aerospace engineering or even mechanical engineering or electrical engineering. So these are their backgrounds. So have a generic undergrad background so that you don't just streamline your because we have to be real. There's no guaranteed jobs always in space sector. So if you have a generic degree, then you can always go back to industries or companies, and then you can transfer your skills there as well. And then once you start going into your master's level or PhD level, if you want to do a PhD, then you can start streamlining your interest into space if you want to do space. There are different courses like even in India and elsewhere, it's opening, I think, more master courses that is focused on space research, as well as in Europe, even like I mentioned about the NEST, which is a research institute in Northumbria. Now they're opening up space courses as well for both undergrad and master. So you can pick your options like that from master. So this is my personal advice usually to the students, where I tell them it's better to streamline from master's level and have a bit more generic science background, because like you saw how astrobiology is fully interdisciplinary. So having a generic background will definitely help. Like if you're an engineer, you can always apply your skills in astrobiology.
I'll say thank you for the answering all the question, and there is a few more questions to go heading before we wind up, dear students. Still, we are opening for the Q&A sessions. What I'm looking for asking you, like while you are just heading with your journey, so like what kind of barriers that you have been facing?
I think in terms of PhD and stuff, I think funding is one of the a big barrier, especially as international students, because most of the fundings are available are for the local students. There's very limited funding, especially in UK, because the international students, even for PhD, still pay very high amount of fees. So if you have to be fully funded, that means they have to compromise a lot of money. So yeah, funding is one of the biggest barriers, but I was luckily I was blessed. I got a good funding for my PhD. And the other background barriers would be, um, I think picking if you want to pursue abroad, it's picking the right country, because a lot of people don't think in terms of long term. Like as long as you're okay to just keep juggling around. But I personally advise students to think in terms of long term. For example, when I went to Switzerland, I know it's a non-English speaking country, but I also knew that after my master's, I'm not going to stay there. So it was okay for me to go. Master's is fully in English, so you can go pursue your master's in everything. But when you're in a non-speaking English country, to find jobs, it would become difficult again, or to apply for funds and then pursue everything would be a bit more trickier. So then you would have to come to a English speaking country or anything like that. So you navigate thinking a bit ahead, not just very time being. So that then otherwise that would become a barrier. Just language would become a barrier for you. A lot of students go to Germany, they pursue everything, but then later they don't get jobs if you don't know B1 level German. Or if you go to Netherlands, it's more or less the same over there. So that's definitely a barrier. And off late, I think for international students, visas has become another big barrier. US is not very happy anymore. Europe is also cutting a lot. So yeah, I think those are one of the biggest challenges right now for international students rather than anything else. And finding a good team as well, which supports you and works with you is very important for you to before you start.
Any kind of students who really are passionate and enthusiastic about the space science, astronomy, and observations, the very first intentions in their brain, it's about the something like space agencies like NASA, ESA, sort of like things, and they were just completely trying to broad their mind over there and trying to something like get some opportunity. So what is your experience and how would you see that case, like not only the NASA, there are lot of space
Yeah, yeah.
institutions and also like in private sector there are some opportunities, and so while you are just seeing the overview of these questions, what is the advice for the young people like where they selecting their future careers?
Oh, yeah, that's a brilliant one. Actually, it's a very, I think, easily misled where they think space is equal to just NASA, ESA, or space missions only. But if you are interested in pursuing an academic career, then there's definitely a lot more opportunities because you can always do space research, tying up with the space agency or not tying up with the space agency. It's fine depending on what kind of research you're doing, because our group is involved in sending instruments to Mars. So they have to be tied up with space agencies and funded by space agencies. That's how they were tied up. But if you are just, say you're a glaciologist and you're just exploring Arctic circles, Antarctica, and you want to understand how the glaciers are, for that, you don't really need space agencies and things. You can just do your research as a glaciologist and, extrapolate those studies to space. Or as a microbiologist, again, you can just work on extreme environments, and then you increase the knowledge through your research about habitability or about life or about how microbes have different mechanisms to adapt to these extreme environments. So you definitely can work in research institutes. You can work in I think even space companies. There are so many space companies as an engineer. If you're computer or hardware engineers, there are a lot of space companies that are working off late as well. AI has become a big part now. So AI is also getting into space sectors, like I mentioned. So if you're interested in computer science, you can always shift anytime to space. I think right now space is very much integrated into not a very niche topic. There's a lot of people can come in and go out. So yeah, you don't have to think only NASA, ESA or anything. You can pursue universities or researchers that are existing and just follow their lead.
Yeah, that sounds very interesting. And also, like, are you looking for starting something like new startup in your own way in the future?
No, definitely. As a biologist, no. I think I like research, so I just want to continue doing research, if possible, in the same field, hopefully. Because as a researcher, it will take time after your PhD to get a permanent position. You'll always keep jumping around as postdocs, as researchers. But hopefully, I'll be continuing research. That's it. I just like to stay in the lab and do lab work. So that's my most interest.
Yeah.
So yeah, we are planning to just do that. But if you guys have any questions about just even like how to pursue these subjects, like or careers, you can always like ask me. It doesn't have to be only about astrobiology or DNA.
Yes, that's what I'm just moving to the last part of the today's session. And also, dear students, I know that most of students are coming from physics background and they maybe like maths and many of that stuff. And I think overall, Jyothi, if you could give some answer for the all the students, like how important to plan in their career guidance and in which stage that they should plan, like starting from school level or university level, because of with my experience in Sri Lankan students, so they are truly interested in Sky as everyone, but unfortunately they don't know how to find out the exact job positions in the future. But same issues available in India, as I know.
Yeah.
And I feel like the very key point that you mentioned, the career guidance for the space science and the astronomy and other subject, what will be the your view for that case?
I think even if you understand from undergrad, it should be enough. In my case, that's what happened. In between undergrad and masters is when I discovered astrobiology. So because your brain would be a bit more mature at that point, and you'll understand your. Of course, by then you have to choose the right undergrad as well. But after your 12th, you'll already identify your strength. If you're pursuing STEM, you'll know whether you're strongest in math or whether you're strongest in science, in physics or in biology. So depending on that, you choose your undergrad program. And then, like I mentioned, when you pick a generic undergrad program and you're getting more interested in space, you can yourself, like attending these talks or a lot of online courses are available, like on Coursera, which some of them are free as well. You can learn more about space. You can just build knowledge. And then for your masters, you can always pursue completely space if you feel like that's where your interest is going to lie and this is exactly what you want to do. Or again, you can choose like some universities even offer like a major and a minor degree. So major credits will be like a generic, for example, life sciences for a biologist. And then minor 30 credits would be like astrobiology if you want to. So having that is also one of advantage. So yeah, you can start thinking more seriously during your undergraduation. And if you're in masters already, also not to worry. You can still streamline your skills to see which kind of group do you fit in. If you're already doing your masters in chemistry, don't think like, oh no, I cannot get into space. If there's any groups, research groups that are working with identification of gases, for example, like I mentioned, or interested in studying organic molecules or things like that, then you can work as a chemist within that group. So anybody, anywhere, even after masters, not to worry.
And thank you for answering all the question. And there is one more questions to go before we wind up, because of
Sure. Yeah.
I also just experienced with some similar question, because of I noticed while I was just trying to providing kind of outreach in Sri Lanka. So I met few people. So they are maybe like around in 50, 40 years old. Like they have been just already passing the 30, but they are still interested in astrobiology and astronomy, because of that was their childhood dream. But unfortunately, they have been just divert their original career to something else. But if someone, not only the Sri Lanka, I mean like wherever, So it's available, still interested in that particular field like astrobiology, astronomy, or whatever subject that they are just dream in the childhood. So if they want to achieve that dream, is it going to be possible, and what kind of career guidance that you are seeing? Because of this is sort of like change, like what we are seeing that usually. Because of I
Yeah,
noticed
it's
even in our group, this group also, just I know that few of my friends, and they also just join over there, and but
okay.
still they do have some DSC and other thing, but they don't know how to just stepping out to the next steps.
Yeah, I mean, from my experience, what I've seen in Aberdeen, we started a master's course there, and we saw some of them were above 50 who enrolled into this course. So you have a lot of part-time options. That's a good thing. You don't have to be like a full-time committed student, because of course after 50 you have responsibilities, you'll have family, so you need to work as well as study, right? So they do these part-time master courses which is related to planetary sciences, and then if they get an opportunity to work, they start like that. But if you think you cannot afford to study or you don't want to study, then you can just, like I mentioned, look into companies where they need your skills. As an electrical engineer or a hardware developer, if this company is working with space instruments or something like that, if you think your skills can go, you can always apply or do some online courses, build your CV, and then always just apply and see if you can pursue. Or yeah, it is still possible. I know studying after a certain age is viewed differently in Asia versus Europe. In Europe, there's no definite age barrier. We've had a student who was actually retired. He's a retired man, but he wanted to not waste his retired life. He was bored and he started pursuing masters, and he was interested in archaeology. So we have these kind of students. But in Asia, normally there's a certain age factor. We say like do not pursue after a certain age or something, because it's mostly we see all of them are more or less the same age in our countries. But I would say if you're open to just studying part-time, go ahead and pursue. If you still want to, you can always navigate different companies, you can always navigate different courses. There's no age for that.
Okay, thank you very much, Dr. Jyothi. And also, like, I would like to ask you something else before we wind up again. And what is the best advice that you ever received?
Best advice. uh, there are many. I
So I
think
think
it's
like
just
you can just share multiple, because of I think like that will be really helpful to some of like our upcoming rising stars over there.
I would say like analyzing your capability is one of the best things. You have to have that skill to know and differentiate reality versus unrealistic expectations and dreams. So that's when you will get disappointed. So somebody used to tell me like know your strength and weaknesses very well, so that you can analyze that if this field is really for me, can I really do this, or should I just do something else? Because a lot of people think space is just it just sounds cool, so I want to be there. It's not like that. There are so many fields in the world that is actually cool. So don't just think like space is the cool thing. So yeah, understand and analyze your strength and weaknesses very well. So once you believe in your strengths, then don't give up on your strength. Just pursue what you can. And if one door closes, like we always hear, the other is always open. So it's just for the best. So yeah, for me, before I moved to Aberdeen for my PhD, I really tried in Edinburgh. It's also in Scotland, and I was on a waiting list for the funding. And the supervisor was very eager to have me on board, and I was very disappointed when I didn't get the funding. And then I got an Aberdeen University. I was not very happy. I was like, where is this Aberdeen? I haven't even heard. It's just 2 hours away from Edinburgh, but I didn't know before anything. But looking back now, that's the team that is holding me right now. They're the one who gave me opportunity to work with them more after PhD right now in Northumbria, and the team is doing a really lot of cool things. So if I would have given up after Edinburgh, then and if I was stubborn that no, Edinburgh is the only place I want to go, then I would lose such a great team. So that's why sometimes you weigh your options. Don't always be streamlined to one direction only. Don't just think if I don't get this, then it's nothing. It's the end of the world. You can always try different things. Always look for opportunities and just grab whatever you get, because in the current economy we have, I honestly don't think our generation has too much options to go and explore. We really need to grab any opportunity we get, because the job market everywhere we hear, not just in India, Sri Lanka, everywhere in the world is pretty bad, including Europe. So many graduates are finding so hard. They're very talented, but there's no job for them. And it's much more difficult for international students. So my best advice is just grab any opportunity you get. No opportunity is a waste. So just hold on to anything you get, and then you can navigate from there. You never know what kind of doors will open. Just navigate from there. So that's what I would say.
And finally, and we have in arrival today our dead end of today's special talk with the Dr. Jyothi Basapathi. I believe like every students over there, they have been just truly enjoyed the sessions, and you are just sharing a lot of knowledge and your experience. And thank you very much
Yeah.
for your time and the devotion,
No,
and
thank
highly
you.
appreciate what you have been done for the whole the South Asian region, because of your trying to upgrading the knowledge of the subject like astrobiology. And I hope like in the near future we can just have another discussions with your upcoming
Yeah, sure.
research. And from the Eco Astronomy, we are just passing you special greetings and wishing you very all the best. And
Thank you so much. Yeah,
yeah, thank you very much again, Jyothi, for participating today's special
no
session.
problem.
Yeah. And
Thank you.
so we are going to wind up from here. Thank you
Yeah,
very
sure.
much, and
Yes.
good night
Thank
for
you.
the
Good
everyone.
night. See ya. See you, everyone. Take care.
So, dear students, we are going to end the program from here. And for the other students, so they do have another sessions from tomorrow, 4 p.m. and Exoplanet students, and also like 6:30 to 8:30 p.m. for the single medium courses as well. And good night for the everyone. And
See you.
yeah, okay, see you. Bye.
See you all. Thank you. Bye bye.
See you and